Kurunji Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
Kurunji Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurunjischoolnkl.com.
Students, now in this video we are discussing continuation information of operand concept. Continuation information of operand concept. So in previous video I have given, so there are two different types of systems. One is inducible and one more is repressible or inhibitable. So now in this particular case, now complete information we are going to discuss regarding inducible system in lac operon of E. coli we are going to discuss. Okay, inducible system. So once you see an inducible operand system normally remains in switched off condition. Means, so what is the inducible system now we are going to discuss. Now this particular inducible system normally how it will be in a switched off condition and begins to work, means normally will be in switched off mode. And suppose if requirement is there, means suppose once you see begins to work. Okay, so suppose if it is switched on, means definitely it starts to work only when the substance is to be metabolized. Suppose if the suppose one particular substance is going to be metabolized, so then only this particular system will be started or begins. Okay, so present in the cell or metabolized by it is present in the cell. Next one, inducible operon system generally occurs in catabolite pathways. Catabolite pathways means, again if you take the metabolism, so there are two different types, so we can be considered. One is anabolism and one more is catabolism. So now the catabolism is nothing but breakdown of complex molecules into simple molecules, or catabolism is nothing but breakdown of substances or the breakdown of molecules, isn't it? Now here inducible operon also, even inducible operon also generally occurs in catabolic pathways. So catabolic pathways a best example only. Now we are taking lac operon concept or lac operon of E. coli. So once you see there, sir, any kind of example you try to give, sir, under active repressor and what is the inducible system, means here best example you see in presence of inducer. In the presence of inducer, entire system will be activated. So means here what is the point? No, once you see active repressor is there. So from this active repressor means I gene is there, no, I gene, regulator gene. So that particular I gene produces the repressor mRNA. So from the repressor mRNA, which one formation takes place? Repressor proteins formation takes place. Now when this active repressor is combined with the inducer, so what is which one formation taking place? Inactivate repressor formation taking place. So means, did you remember one point? So this particular point we have learned in presence of lactose in the cell, isn't it? So means actually repressor mRNA produces the repressor protein. So now the repressor mRNA or the protein is active, but when the lactose is acting as an inducer, no, this particular lactose will go and binds to the repressor. Automatically the activity of the repressor protein is inactivated. So that is the best example here we are taking. So means here inducible operand concept or the inducible operand system generally will be in switched off mode, but it works only when the substance to be metabolized. So when one particular substance is going to be metabolized in one reaction or in the cell, then only this particular inducible operand will be started. Okay, so means here inducible operand system generally occurs in catabolytic pathway. Why? Because here once you see this particular inducible operand begins to work when the substance to be metabolized. So means here one particular substance, so for example if you take one lactose, that particular substance if you consider that will be metabolized. So here, so what are the kinds of pathways are going to be taking place? Catabolic pathways. Okay, so now here catabolic pathways, best example here we will be considering as Lac-operon of E. coli. Okay, now here if you take the structure of Lac-operon of E. coli now, so that will be like this. So there are different types of genes, so together we will be considering. Okay, now here promoter gene is there, operator gene is there. So promoter regulator gene and promoter operator gene and ZYA, these three are structural genes. So this one, so we know very well in in prokaryotes, same this kind of structures means polycystonic conditions only we will observe. So best example PIPO-ZYA. Okay, now if you take one inducible operand system, if you take one particular inducible operand system, that particular inducible operand system consists of 4 types of genes. So how many? 4 types of genes. Once you see, so what are the 4 types of genes we will find out means, first one let us start to take structural genes. Okay, structural genes. So these genes synthesizes. Okay, these genes. So these genes synthesizes the so now you see these genes synthesizes mRNAs. So means definitely here the structural genes means here ZYA genes together we are considering as structural genes. No, so now these genes synthesizes the mRNAs. So which in turn synthesizes the polypeptide chains or enzymes. So means here first of all by the process of transcription and by the process of translation. So here by the process of transcription mRNA formation takes place, and by the process of translation polypeptide chains of formation takes place or enzymes of formation takes place. So now here definitely when we are observing the structural genes, so here definitely we will find out we will observe the presence of 3 different types of structural genes. So now here if you move forward to learn now, an operand may have 1 or more structural genes. Correct only. Now suppose if you take one particular operand model, there 1 or more structural genes can be present. Okay, definitely are present. That's why 1 or more structural genes are present. Now here in Lac-operon model of E. coli, how many number of structural genes are there? 3. One is Z, second one is Y, and third one is A. ZYA genes, isn't it? Right. Now each structural gene of a operon, so each structural gene of a operon is known as a cistron. Means here functional unit, no. So cistron is a functional unit or the functional unit of inheritance, isn't it? So that's why here each and every functional gene we can be taken as cistron. Means here Z gene as one cistron and Y gene as one cistron and A gene as one cistron. That's why here in prokaryotes what is the kind of condition we are observing means polycistronic condition. Polycistronic means here more than 1 or more than 2 number of cistrons are there. That's why polycistronic condition we will observe. So now under structural genes, what are the main important points we discussed? So structural genes definitely involves in the synthesis of mRNA, so which in turn gives polypeptide chains or enzymes. So means here the polypeptide chains or enzymes so produces the different types of So here the polypeptide chain definitely produces the different types of enzymes. So enzymes names and everything. So here down we will observe. Okay, now here each structural gene if you see, there no, and in one operon each structural gene we will be considering as cistrone. Next one, lac operon, lactose operon. For example, if you see there no lac operon, so for example simply lac operon we will be considering as lactose operon of E. coli contains 3 structural genes. So already we discussed, contains 3 structural genes, for example ZYA genes, and these genes occur adjacent to each other. So again sir, here these 3 structural genes are there. Now how they have been arranged, sir, means so these 3 structural genes are situated or lies beside or adjacent to each other, thus and are linked. Okay, and they have situated very close to each other, and they have been situated beside or adjacent to each other, and they have been linked. Okay, next one, they transcribe a polycistronic mRNA. This is important point. So means here we are studying there are 3 structural genes, ZYO genes are there, and these 3 structural genes definitely transcribe the polycistronic mRNA. So means here if you take one, that's why here polycistronic gene which is synthesized in prokaryotes we have taken, we studied. So thereby here what is the point we can understand, if you take these 3 structural genes definitely involved in the synthesis of or definitely involved in the formation of polycistronic mRNA. Polycistronic mRNA. Okay, and this particular mRNA molecule that helps in the synthesis of 3 different types of enzymes. Correct only no. So means here suppose in presence of lactose what is happening, definitely these 3 structural genes, so for example ZYA genes will produce the single stranded mRNA by the process of transcription, later by the process of translation. So here there are 3 different types of enzymes production takes place. So these 3 different types of enzymes from where will be produced, means beta galactosidase is produced from the Z gene, and what is the use of this beta galactosidase, means that helps in so breaking of lactose into glucose and galactose, or simply that breaks the glucose or that breaks the lactose into glucose and galactose. So thereby here 2 important functions we can observe the beta galactosidase. Okay, so here one function you can remember, that is this glucose that breaks the lactose into glucose and galactose. Next one, second enzyme if you take, then there no, that one is permease enzyme. So then what is the use of this permease enzyme, means that definitely that helps in entry. So that helps in the entry of lactose in cell from outside. So means here definitely permease helps in the transport. Already we have discussed this particular information. There are initially one more time I'm saying, listen carefully. So in structural genes, 3 different types of structural genes are there. So these 3 structural genes are nothing but ZYA genes, and these 3 ZYA genes produces 3 different types of enzymes. So there are 3 different types of enzymes over there. So one is beta galactosidase enzyme. So what is the use of a beta galactosidase enzyme, means that breaks the lactose into glucose and galactose, and second gene of a structural genes Y is there, that produces the permease enzyme. Then what is the use of this permease, means definitely it involves in the entry of lactose into the cell. From where? From outside of the medium. Next, or from outside of the cell or from the outside of the organism. Next one, transacetylase. Third one. So this particular transacetylase enzyme produced from the A structural gene. A structural gene. Then what is the use of this transacetylase enzyme means? So that transfers, actually in our textbook, so they said that there is no the function of transacetylase, likewise they have given, but here in another book now we are referring transacetylase transfers an acetyl group from acetyl CoA. Means once you see it transfers an acetyl group. So means here this transacetylase enzyme which helps in the transfer of an acetyl group from acetyl CoA to beta galactosidase. So means here transfer of acetyl group we are taking. So that's why here we are taking no transacetylase means trans means transfer. So which one is going to be transferred? Acetyl group. So that's why here what is the work of this particular transacetylase enzyme means that it transfers an acetyl group from acetyl CoA to beta galactosidase. So these three differentially, so we will be considering as so three different types of functions of three different enzymes respectively. Right. Now operon gene, second one. So second one, if you take that is the name is operon gene, it lies adjacent to the structural genes. Correct only, no? Again, once if we see the structure, this particular operon is there, no? So in a operon only, so operator we are taking, isn't it? So this particular operator, it lies adjacent to the structural genes. Correct only, no? So adjacent na pakkatalem. So it is situated beside to the structural genes and directly controls the synthesis of mRNA over the structural genes. Means here, so here one more time you read this sentence, directly controls the synthesis of mRNA over structural genes. Means here mRNA formation, so from the structural genes. Suppose if the structural genes would like to produce the mRNA means, so that particular process definitely depends on the operon. Why? Because here this particular operon only playing an important role in regulation. Suppose if this operon is binded by the repressor mRNA or the repressor protein means these three structural genes will not be transcribed and will not be translated furtherly. Okay, so thereby here what is the point? So this particular operon directly controls the synthesis of mRNA over the structure. Okay, next one is it is switched off. So generally operator gene that will be switched off by the presence of repressor. So correct only, no? So actually regulator gene is there here, I gene. So this particular I gene produces the repressor mRNA. So that particular repressor mRNA produces the repressor protein. So when this repressor protein will go and binds to the operator at the time, so the operator's gene function will be switched off. Why? So here the repressor will go and binds to the operator. So next one, an inducer can take away the repressor. Means here even though suppose here repressor is there, so repressor work one and only can change, adi an inducer. So that's why here we have written like this, an inducer can take away the repressor. So means here one repressor is there, generally in absence of lactose this repressor will go and binds to the operator, automatically operator work will be switched off. So when work of operator gene or operator gene is switched off, no structural genes will not be transcribed. So thereby here what is the thing here we are observing, no? So suppose in presence of inducer, suppose if the repressor is taken away from the particular situation means at the time only switch on the gene that directs the structural genes to transcribe. So means in absence of repressor protein in the operon model, means here when the repressor protein when the repressor protein cannot binds to the operator, then only entire mechanism will be switched on. So that's why here the switched off mechanism or the switch on mechanism, everything definitely depends on the operator region. Why? Because suppose if the operator region binded by the repressor protein means at the time operator work will be switched off. Suppose if the operator region not binded by the operon protein means at the time only we will observe the switched on mechanism of operon gene. So suppose if the operator gene is in a switched on mode means then only structural genes will be transcribed. Next gene is promoter gene. Next one is a promoter gene. This gene is a site for initial binding site of RNA polymerase. So means here the promoter region is there. That particular promoter region is acting as initial binding site. Means initial binding site. First of all, initial means first one. So initially RNA polymerase enzyme or DNA dependent RNA polymerase enzyme. Okay, so here definitely binds to the promoter region. So when this particular promoter region or this one binded by the which one? So here you take otherwise here. Okay, so here if you take the binding site of RNA polymerase enzyme. So initially RNA polymerase enzyme will come and binds at the promoter region. So when operator gene is turned on. So means turned on means when we can observe this particular situation when operator. So when this particular operator gene is switched on, when the operator gene is switched on, once you see when the operator gene is turned on, so means here operator gene is turned on means here repressor protein which is obtained or which is synthesized from the I gene is not binded to the operator. So then only we will observe the switched on mechanism of operator. So at the time the enzyme RNA polymerase enzyme, the enzyme which is RNA polymerase enzyme, that particular RNA polymerase enzyme moves over it and transcribes or reaches the structural genes. A point you understood the means. Suppose here what is a RNA polymerase enzyme present at the promoter region, that must move towards the structural genes. So then only when this RNA polymerase enzyme present at the promoter region, when it moves towards the structural genes, then only these three structural genes are transcribed and finally proteins formation and enzymes formation takes place. So suppose here this particular operator gene is bounded by or blocked by the repressor protein means at the time the mRNA or RNA polymerase enzyme cannot overcome the operator region. Why? Because here operator region is blocked, isn't it? So thereby here what is the point we are discussing? When the operon gene is turned on, the enzyme RNA polymerase enzyme moves over it and reaches the structural genes to perform the transcription process. Next one, regulator gene. So now what is the use of this regulator gene? Now let us see. It produces a repressor. So regulator gene, it produces a repressor that binds to operator gene. One more time, regulator gene, it produces a repressor. So thereby here regulator gene is nothing but I gene. So in brackets we can keep so here actually this entire information even helps to your public examination also. Suppose if they ask detailed information of lac-operon module means, so at that time each and every point what is the information now I have written on the board, every point you can explain. Okay, structural genes. So here we have taken now structural genes, operator gene and promoter gene and regulator gene and repressor. Next one, even inducer also information is there. Now regulator gene point of view, it produces a repressor that binds to the operator. So that's we know very well. Repressor protein is there means definitely that particular repressor protein or repressor repressor protein binds to the operator gene and stops the working of operator gene. So stops the work of operator gene means definitely operator region will be switched off. So that's why here we have kept name it as regulator gene. Next one, repressor. Now here it is a protein. So repressor, it is a protein produced by the regulator gene. So this particular regulator gene involved in the synthesis of repressor protein. So this repressor protein binds to the operator gene so that the transcription of structural genes stops. Means here the transcription of structural genes mainly depends on the regulator gene. Suppose so here this particular regulator or repressor protein will go and binds to the operator, so entire process will be stopped. When this repressor protein cannot be bind to the operator, at the time the pathway of RNA polymerase is blocked. Okay, so at the time the pathway of RNA polymerase is free. Means here once you see here RNA polymerase enzyme is there, and suppose here this particular operator blocked by the repressor protein means at the time this RNA polymerase enzyme cannot come like this. But here repressor protein cannot be binded at the operator region means now the pathway of RNA polymerase will be free only. At the time definitely entire mechanism will be switched on. Okay, next one, last and final point, that is repressor protein or the repressor has two allosteric sites. One is operator site and second one is effective molecule, that is inducer or co-repressor. Okay, two allosteric sites also we will observe in repressor. One is operator site and one more is effective molecule, that is maybe inducer or co-repressor. Now here we have written or we have drawn, now you see in absence of lactose, so here first diagram that indicates, that gives information in absence of lactose what is happening. So we can study from this diagram. Now you see in absence of lactose, so from the regulator gene, I gene is nothing but regulator gene only. From the I gene repressor mRNA formation takes place. So in the entire sequence PIPO-ZYA. Again I am not going to write P means promoter, I means regulator gene and O means operator, ZYA genes or structural genes. I am not going to write. Why? Because we know very well. Okay. Now in absence of lactose, what is happening? Regulator gene produces the repressor mRNA and by the process of transcription, later by the process of translation repressor mRNA. So 0. So it is nothing but repressor protein. So this repressor protein will go and binds at the operator region. Suppose if this repressor protein will go and binds to the operator region means at the time our knowledge point of view, promoter region definitely contains or provides a binding site for the RNA polymerase enzyme. If RNA polymerase enzyme is there at the promoter region, exactly at spotlight. But here RNA polymerase enzyme definitely must go towards the structural genes to transcribe these three structural genes. But here when repressor protein is blocked the pathway of RNA polymerase enzyme, that cannot overcome this operator region. Why? Because due to the presence of repressor protein. So that's why here from the structural genes we could not see the formation of different types of genes or the different types of proteins. So formation no chance. But in presence of lactose, so in presence of lactose is here acting as inducer. Lactose is acting as inducer. Again same sequence. Now PIPO-ZYA we took. Now from the regulator gene by the process of transcription mRNA means repressor mRNA. The line repressor mRNA or line indicates repressor mRNA. From the repressor mRNA to which one formation we are observing? Repressor protein. Now till now repressor protein is active only. But when inducer means inducer is nothing but lactose, no, in presence of lactose, so inducer or the lactose will go and binds to the repressor protein. At the time, no, the activity or the structural functional systems of the repressor protein is inactivated when the lactose will go and binds to the repressor protein. So actually the normal function of the repressor protein will go and binds to the operator, but in presence of lactose, when this particular lactose acting as inducer, so it will go and changes the functional activities. So at the time, no, this particular repressor mRNA or repressor protein will be inactivated and cannot be binded to the or cannot be binds to the or cannot be bounded to the which one operator. So that's why here we have kept into mark. So in this particular case, no, repressor protein cannot be or cannot binds to the operator. So at the time you see promoter region already consists of RNA polymerase enzyme. No, now here the pathway of RNA polymerase is free. So thereby RNA polymerase will move towards the structural genes, and by the process of transcription single stranded mRNA formation takes place, and by the process of translation 3 types of proteins, for example from the Z gene to beta galactosidase enzyme, from the Y gene to permease enzyme, and from the A gene to transacetylase enzyme formation takes place. So here I don't have a place to write down the 3 different types of enzymes, but through my voice I have given. So here this particular diagram indicates in presence of lactose what is happening, and here in absence of lactose what is happening. In left side diagram, no, we have discussed. So this is just somewhat information only. So if a possibility is there, so one important concept which is not given in our NCERT, even that also I will explain in the next class. So until then, watch this video 1 or 2 times, so gain the knowledge. Suppose if you learn this knowledge, no, just listen this by 1 more time, you will be revised. So this entire information will be revised, so thereby for the public examination preparation point of view, no, so you will get this entire information so within a short period of time, including absence and presence of lactose what is happening. But for NEET examination point of view, one more small concept I have to give, so that particular information in the next video I will try to give. Thank you and be safe at home.